Today we're speaking with Dr. Svetlana Mojsov, a biochemist and peptide chemist at Rockefeller University and then Mass General, whose work was central to predicting, synthesizing, and demonstrating the biological activity of GLP-1, a molecule now at the heart of a global therapeutic revolution.
Dr. Mojsov has received some of the most prestigious honors in science, among them the Lasker Award, the Breakthrough Prize, the Tang Prize, and most recently the King Faisal Prize for Medicine. And she was also named one of Time magazine's most influential people in 2024, and we are really delighted that she joined us.
We're so excited to meet you.
Yeah, thank you. Well, thank you for inviting me.
I will give you a little bit of background on why we are doing this.
Mm-hmm.
I am Ani Deshpande. I am a cancer researcher in La Jolla, and Pam is diabetes and pancreatic cancer researcher. And we do our own writing grants and publishing papers and all that. But I thought that there was a strong need to communicate science effectively, because despite all the big breakthroughs in medicine, people do not appreciate the work that goes into all the things that scientists do. And so we wanted to tell the stories in a way that they are both entertaining and informative for the lay public. So we tried to keep it as simple for the lay audience as possible so that they can appreciate the enormous effort that go into the discoveries that are made in medical science. So that's the general overview of the project. I don't know if you got to see some of our work.
Yes, I did. It was very helpful just because I realized actually it did say it had a message, and I realized what the message is, and that's why. And I also appreciated that you actually read my papers. It was to pull out my name out of the hat. And so I knew that there was you had the scientific background.
Yeah. I mean, in some ways, we'd love to have a more scientific discussion, but our goal is to help other people appreciate how all these advances are saving their lives. So maybe I'll start with the first question. I was so struck that you were born in Yugoslavia and got a bachelor's degree in physical chemistry. And I remember how difficult physical chemistry was. And then you traveled halfway around the world to New York and earned a doctorate in protein chemistry. Both of those fields were pretty esoteric at the time, and there were almost no women in either one. So it made us wonder, were there moments that made you realize the importance of the fields or inspired you and set you on those paths?
Well, the main point is that growing up in Yugoslavia, the educational system was such that we were not taught that there are women's and men's profession. Women were encouraged by our professors, middle school, high school, to really pursue what we are interested in. And so growing up there, I did not know that there is a woman's profession. I was introduced to chemistry in 6th grade and physics, and I just followed up my interest. As a matter of fact, my classmates in the Department of Physical Chemistry, when we started in the late 60s, were 50% women. And actually all of them became very successful professionals, whether in academia or in whatever they chose to do, physical chemistry.
What's striking here is Mojsov's intellectual trajectory from physical chemistry in Yugoslavia to protein chemistry at Rockefeller, and then molecular biology, a path that positioned her almost uniquely to recognize what others had overlooked.
Now, I came here because physical chemistry was very strict program. You just learned physics and chemistry. And I realized that I didn't really learn any biology. It was the 70s, molecular biology was exploding. And when I told to my professors that I want to do something which was to learn biology, they say, well, go to graduate school in England or United States. And that was not unusual. There were a lot of fellowships which were available for whoever wanted to pursue academic science, at least in my department. So I'm talking about my department of physical chemistry. And a lot of them came, actually, Fulbright fellows. So there was nothing exceptional about me coming to United States, to Rockefeller University. It's just that one of professors says, why don't you go to Rockefeller University? I applied and I was accepted.
At the time, Mojsov's doctoral work under Bruce Merrifield, who would go on to win the Nobel Prize for solid phase peptide synthesis, positioned her at the frontier of a method that was transforming biochemistry.
Great. So forward thinking at the time.
That's right. And I must say there were, at the time, the class of 1972 was 5 women and 25 men, but there was never, at Rockefeller University, but there was never anything saying that we were not in our own profession. There was a big support. The graduate school was very supportive. So I don't see that I never thought I did anything unusual.
Ah, okay. Well, probably you have done a few things unusual, and we're going to get to that. Okay.
So, in our podcast, we have talked a lot initially in the first few episodes about insulin, but we are only beginning now in the newer episodes to talk about other hormones that play important roles in sugar, such as glucagon. And one of your major achievements initially, even as a trainee, was streamlining the process of synthesizing glucagon. And so, could you please tell our audience why this was important?
So this was the 70s, and it became obvious that glucagon plays really a critical role in controlling glucose metabolism. And there was a whole, actually, suggestion, which was published by quite well-known scientists, Unger and Orci, which they even implicated glucagon in the pathophysiology of diabetes. And so in our thinking in Merrifield's lab, we thought that, well, it's possible that if we develop an inhibitor of glucagon, something which will suppress glucose, and it will be administered together with insulin, we will come up with a new and better treatment for diabetes. So that was the motivating force. Found it even then, in the beginning of the 1970s, to develop a drug for diabetes. Merrifield was very much part of it. I think I was influenced by that. And the solid phase was the method to go. You could just have a single scientist do it by himself. But the synthesis was very difficult for glucagon, and I had to devise different synthetic strategies to streamline it, as you mentioned. And we were also encouraged in 1977 or 8, NIH had an announcement asking for grant proposals, which will lead to understanding diabetes and to maybe even coming up with new medicines for diabetes. So Dr. Merrifield and I applied. Dr. Merrifield was the principal investigator. I was the co-principal, and we got a grant. And so that was very stimulating because the NIH, which was also kind of supporting our ideas. And of course, as a graduate student, what else could I have wished for? And actually, by the way, Dr. Merrifield kept that grant for the next 30 years. And he really wanted to develop that inhibitor. And with the synthesis which developed, with the streamlined synthesis, they obtained over 200 glucagon analogs, which was really something which we couldn't have even anticipated in the 1970s. And so it just showed the power of solid phase, the power of forward thinking.
It's worth noting the field into which Mojsov was moving. The incretin hypothesis had been debated for decades, but the tools to test it rigorously had only recently arrived.
And I think there was the scientific society, the scientific environment was very supportive.
That's excellent. But for listeners who may not know the history, one of the great mysteries in endocrinology was called the incretin effect. And can you explain for listeners what that is and why it hinted that the gut was secretly orchestrating much of our insulin response, even before anyone knew about GLP-1?
Well, the idea that the gut can influence the secretions of the pancreas goes back 100 years ago to the beginning of the 20th century. And it was revisited especially in the 1930s, where the name the incretin was coined. It was standard for insulin secretagogue. And incretin. And but the methods were not there, and the results are unreproducible until the mid-1960s. And I think there was a major breakthrough by Rosalind Yellow and Bernson, who developed the first radial assay for insulin. And then he started answering questions. And one of the first experiments was done in the mid-1960s, when it was shown that it was a clinical study, a very small one. It was shown that if glucose is taken orally by mouth, there was a higher levels of insulin compared when it was administered intravenously. And that immediately suggested there must be peptides which are secreted in the intestines when we eat a meal. And then in turn, they will stimulate insulin release from the pancreas and will impact glucose metabolism. So it was those experiments which really proved that. Then it became clear immediately that incretins can be used as new medicines to treat diabetes. And that's why there was a really big effort starting in the 1970s to discover the incretins.
That's wonderful. So now when glucagon like peptide 1 or GLP-1 was found, it generated even at that time a lot of excitement, but the initial experiments actually failed. And we would like to hear in your words, what was the critical thing that you realized?
I want to show you this slide because this is my really raw notebook, and I think it will help to show, and then we can show another one. Okay, so the first paper was published by Graham Bell in 1983, and I was really deeply involved in glucagon biology, biochemistry. I knew its sequence backwards and forward. And actually, I was writing my second paper describing the improved synthesis of glucagon. So glucagon was it's in my mind. And so when I saw this sequence, as I showed, and this is really my rough data, I was so familiar with the glucagon sequence, I knew immediately that the sequence homology of glucagon will start at this position 7. Because, and I knew and it went on. And I knew from my own work how important that histidine at the amino terminus was important for this biological activity. I did that experiment with glucagon. If I removed the histidine, glucagon became inactive. 1 amino acid difference. And I also noticed that before that histidine, there is an arginine amino acid, which is typically cleaved by enzymes, tryptic-like enzymes. And we can also see here how I was counting where the active sequence starts. You see 1, 2, 3. I was trying to see where there is a similarity to the glucagon sequence. So if you see, I actually put the phenylalanine, I put a star, because I knew that glucagon had phenylalanine at position 6. But then nothing else made sense. Then it came to arginine. So this was really my notes. And that's where I predicted that it's going to start at histidine and it's going to be the biologically active peptide. And if you go all the way to the end, you can see how I made a mistake. I put the arrow next to the 2 arginine amino acids, which I knew it has to be cleaved, and I moved it back. So this is the row, and I said glycine, and I put here GLP-1 end. And so the star is, so this arrow next to arginine was put, this is my thinking. The star actually, it was the graphic artist who put it in order to help my presentation. So this is new, the last. Yeah. So it's very clear. So this is it. And I realized immediately this was going to be biologically active peptide. And I started immediately, I think, this came out in in July, the sequence of 1983. By September, the dates in my notebook show I start making the antibodies because I realized immediately. So the experimental challenge was really how to show the GLP-1 is secreted in the intestine. And trying to isolate it was not an option. I knew that I have to just design methods which will allow me to detect it. And that's how I came. First was to actually make synthetic peptides, to obtain antibodies, to raise antibodies in rabbits, to develop radioimmunoassay, which by then was really which was established. It was almost 20 years in use. And so it was very quick. It was kind of straightforward. Even when I read my notes, it was just one step after the other. I must say I was also very fortunate that at that time I met George Cahill, who was the head of Howard Hughes Medical Institute, and who interviewed me to become the head of the proteomic facility, the peptide synthesis facility at the Mass General. And I told him what I was doing, and he said, go ahead, as long as you can manage both, we will support you. So I got a financial support on the Howard Hughes Medical Institute, so I didn't have to write grants. I didn't have to justify, because it was really just an idea. It was a hypothesis. And then just going back to your question, so in 1984, Stephen Bloom's lab published that GLP-1 was not 1 to 37, the long one. I took it as a good news because my hypothesis that the big one, that all the 37 amino acids is not going to be active. This charged amino acids are unimportant. And so I think it actually helped me because I think it slowed down the field. Because he said, it's how glucagon-like is glucagon-like peptide 1. It's not in glucose metabolism. Yeah, and it gave me a little bit more of a breathing space because I did work along with the technician. So I was not disappointed. It was the opposite.
Isn't that funny? You know, I want to say for the audience, we told you when Ani and I saw this figure, we both had chills, and we each have things we've saved from our research. I remember the first time I sequenced a gene. I still have the radio from it, the giant gels, and it's so perfect that it's in your own handwriting. So thank you for showing it.
What follows is a description of the first double-blind experiment in which GLP-1 was tested in a perfused rat pancreas, a moment that Mojsov still remembers vividly decades later.
And was there a particular moment then after this when you realized this peptide might change diabetes treatment?
Well, the idea was that this would be the incretin. And so when I detected in the intestines, it was exciting. It was there. But it was really the first studies in the perfused rat pancreas with Warren Triennial. We were cautiously optimistic. And Gordon Weir was really very generous. And I went to his lab and worked with his research assistant. And it was a double-blind study. So it was Diana Deary who will do the surgical experiment. It was really quite tricky. And then we'll collect samples, we will divide them in half. Half of them will stay, and it was supposed to be a double-blind study, and half of them will stay at the Jocelyn, and they will analyze it for insulin, and half of them I will take it to the Mass General, and I'm going to analyze it for GLP-1. And we said that we are not going to exchange it until we had the data. And I sent the data, and before I could even realize, Gordon Weir called me on the phone. And he said, you won't believe it, how active it is. This was 10 to the minus 11 more. He says, I've never seen anything like that. And then we did a lot of studies to confirm it, but this was very exciting. I actually saw him recently at an event at the Mass General, and he still remembers that moment. I think he had that moment first. And he remembers how he called me, and he called Dr. Habner then because Dr. Habner introduced us. He arranged for that experiment. So that was the first one. And I think it was Gordon Weir who said, well, now you have to go into clinical studies. You have to put it in patients. I think that was Gordon's next. And so the second point was after we analyzed the data from the clinical studies with David Nathan. And again, double-blinded study, but we analyzed both insulin, which at that time was commercially available, and GLP-1 in my lab with my research assistant. And David's lab was analyzing the glucose levels.
This is where things begin to shift from an experimental peptide to something both researchers could feel in real time was going to change medicine.
And I remember we didn't know what it is, but the way we arranged them in the gamma counter, we will just put insulin and then GLP-1, and it was clear. Every single individual responded. It was clear who got and who didn't. Actually, but they were standing. We were really standing in front of the gamma counter watching it. And I remember it was Alice Allen, who was research assistant at that time, and you look at the other and said, that's going to be the drug. So it was really true. And David, at the same event now at the Mass General, David told me, he says, I almost thought that you broke the code. Because it was just incredible how because usually in clinical studies, you don't expect every individual to respond. No. Oh my. And so and then of course we did much more. And so I think at that time I was convinced. And I had a lot of people around me whom I even told this is going to be a drug. And they will all say, oh, really? Okay, all right. But I really believe that because it was just too good to be dropped. Of course, there were problems then, like short half-life and all of it. Sometimes I think maybe it was just an excitement of a young scientist to see. Maybe it was a little bit unrealistic. Now, when I read how difficult it was for Novo Nordisk to develop them and all of it, I said, oh gosh, they really stayed with that. They could have dropped. Yeah.
But I think for all of us, being around our trainees that aren't weighed down by knowing all the problems that are going to come later is very helpful to keep that enthusiasm going, right?
I must say, there was one thing which I learned from Dr. Merrifield as a graduate student. He always told us, you always have to do your own experiments because you will know what works and what it doesn't work. And I think that was probably, in my case, this was the example because I did all the experiments. And when you do the experiments, you think about it, right? You've done this, you know what it is. And it's probably just because it was my experiments that I realized that it's actually going to work.
In the broader context, what made Mojsov's contribution distinctive was not just the prediction. It was the end-to-end execution, from sequence recognition to synthetic peptide, to antibody, to radioimmunoassay, and finally, to clinical sample analysis.
Say that you made it sound so simple, the seven peptide. But when we read about this, I was telling Pam, we were just astonished that right from the prediction to making the peptides yourself to making all the antibodies yourself, we read about the rabbits and how you had to take showers sometimes.
Yeah, 22 rabbits.
Then making antibodies, all those antibodies at a time when it was not trivial, and then using them in those assays to look at the presence of the GLP-1 in the gut. All of that is just any one of that achievements for us would have.
Oh, yeah. It's funny too, because we're talking about Yallo developing radioimmunoassays, and then John Eng, who was Gila Monster, is from her lab.
That's right. I was about to say for one of your questions, one of the most amazing, I think, developments in GLP-1 is John Eng. And you're right that it came, I didn't connect it yet. It comes from Rosalind Yallows. Actually, something which made me also very excited, when I first presented this data on one of the clinical meetings, Rosalind Yallow was sitting in the first. And he actually came and said, you did a good job. That was really very inspiring for me. Of course.
That's actually super interesting because, in our next episode that we are making, which is actually on metformin, we're talking about how important it was, Rosalind Yallow's method development.
Yes.
And then, of course, we will talk about GLP-1. But these are wonderful things.
Oh, thank you.
At the time, John Eng's discovery of exendin 4 in the Gila monster, which he pursued independently, largely without institutional support, reflected the same prepared mind quality that Mojsov describes here. when I read about John Eng, first I was wondering why would anybody study a peptide from a lizard? And then, when I read that he trained with Rosalind Yallo, it all came back, like Pasteur said, that you have to have a prepared mind.
Yeah, exactly. And that he realized so quickly that it was so similar to the sequence of 7 to 37. It was very quick, and that he put it into mice. I think that's a tremendous story. I agree. And also that Amylin, that a small pharmaceutical company, took the risk.
Exactly, when no one else was interested, and he had to pay to develop their own patents.
Because that's right. Because none of these pharmaceutical industries were interested in that. And it was the first injectable GLP-1 analog, first injectable new drug for treatment of diabetes. For 5 years, it was the only one. I very often in my talk say, I think this really broke the ceiling, because there was this belief that peptides cannot become drugs because they have to be injectable. And he and the amylin, they just moved through it, and it showed that if you need to improve your health, you will inject yourself. And especially it was surprising that this was with insulin. They were all injected. They were used to it. So it was really very close-minded pharmaceutical company, and John broke through it. I think that's a remarkable accomplishment.
So I want to ask you, around the same time that your group was discovering GLP-1, there were researchers on the other side of the world, or other side of the ocean, that were uncovering similar peptides through different approaches. So what was it like to see the research efforts converge, multiple groups arriving at similar conclusions through different directions?
Well, that was always really very reassuring. One thing which I would like to point out was really Stephen Bloom, right? He first was wrong. But then he came at the end of 1987 with a clinical study which showed that GLP-1 is the incretin in men. Big breakthrough. And that was very reassuring to us because it really confirmed our belief. And by the time that paper was published, we were already thinking about going into clinical study. And that was very reassuring, I think, probably to David Nathan too. And because it showed that it was effective in men, that it's an incretin, and that the only thing left now to see how individuals with type 2 diabetes will reflect. So for me, that was the most encouraging and important paper which came. And it was good for the Bloom's group that they kind of revisited.
Yeah.
What's striking here is how the field converged from multiple directions: molecular biology, classical peptide isolation, clinical physiology, each approach incomplete on its own, but each confirming the others.
And now, even more astonishingly, GLP-1 drugs seems to be implicated in cardiovascular health, kidney function, and maybe even longevity. What are your thoughts on that?
Well, I think that it just shows that some homeostasis have to exist for good health. And I guess, these peptides and GLP-1 among them, that's what their function is, to not only control glucose metabolism, but to actually control all the different aspects of it. And that's cardiovascular and even kidneys. It was known that actually glucagon also has at that I knew even as a graduate student, also has effect in the heart. And it was given for years in patients to speed up their heartbeat that they were actually at the emergency room.
I've never heard that.
And also that was known. And then it was just something you did. Nobody was thinking in terms of mechanism and networks. So I did an experiment in the 1960s with a colleague, Yang Wei, in which we looked for the expression of GLP-1 receptors in different organs. And we found it in the heart. And I must say, I did think of glucagon again and how it had a therapeutic value in some very limited cases. And so they didn't follow up. It was really Novo Nordisk who was required to do that. But that somehow put the big picture together. The kidneys, I think, yes, it's overall homeostatic balance in the body to keep us healthy.
I just find it fascinating beyond and above what you just described, the effects of this hormone on the brain. I find it astonishing, the studies that are showing that it broadly influences satiety and cravings and maybe even reward pathways associated generally with addiction. So I would like to hear your thoughts on that.
Well, I think that's fascinating. I think that's the new frontier. And I think this is really what helped with these long-acting drugs like Ozempic is about, because it showed us that there are other pathways which can be regulated by peptides, GLP-1, and I think probably now there will be others. And so I think it's very important that we understand that. So that's what I'm the most exciting, how GLP-1 regulates addiction, but the whole rewarding behavior, the food noise. Apparently, there are also the reports that it influences addiction, nicotine addiction. So I think GLP-1 will now give us a handle to understand what it does in the brain with this new long-acting medicines, because we knew that there was GLP-1 in the brain. That was shown that it was in the brainstem. But they had no ways to even try to understand what it does, and now we know. Now I almost think we have set up what kind of experiments we should do in order to understand that and to develop therapeutic interventions to actually take care of it.
I mean, what a godsend for people if it really helps with addictions to everything.
And the weight loss is very significant because all the individuals will say how much better they feel when they lose their 100 pounds. Because that's a big burden on the whole organism. And I think that, again, was not anticipated. It really came from the scientists at Novo Nordisk. But I think that's a big insight.
Yeah, and I believe that people who are obese who take the drug lose more weight even than diabetic patients.
That's right. Much more. And so we have to also understand why that's happening. Yeah. Of course, the doses are being different, but still, I think the effects are much more profound.
So you've had the rare experience of seeing our basic research go from the bench to the bedside and, as a blockbuster drug. What is it that you still find intriguing?
Well, the effects in the brain, right? That's what is most intriguing now. And that was not anticipated. I keep saying in many of my talks, when we were discovering incretins, we knew what we were looking for. I knew what I was looking for. And so and you said it was one experiment after another. It was kind of obvious. But now with the brain, we don't know what you're looking for. We go back. It was really the clinical experience which opened a whole new field for us to explore. And I think this is the beauty of science, right? You solve one problem and then the other one comes. And there's the challenge. What's next? And I think that's the challenge which we are facing. Thank you very much that you contacted me and that I can talk about my work. Thank you. Bye.
Thank you for listening to this Discovery Dialogues Deep Dive. Stay tuned for more conversations with scientists whose work has shaped how we understand and treat disease.